In this section, we're going to discuss the tabular results page from the protein family sorter. Last time we left off, we had added some genomes to the group. We had selected protein families. Now we hit the search button, and that launches the tool. It rewrites the current window to show that. You'll notice that on the left-hand side, there is a dynamic filter. It says filter by, and it's set at genus-specific families, but you could change this at any time to global families. If I pull this over, you can see that you have the different genomes here. It starts listing them in alphabetical order, but there's different data available too that you can sort on if you so desire. Let's go look at the tabular results. So first, in the first column, you have the ability to click on the individual protein families, but they start with the numerically, it starts sorting the table by this unique identifier for this particular protein family. This particular protein family, the functional name assigned to it, it includes mobile element proteins that all have the same k-mer. Within this family, and across these 26 of the 39 genomes, there are 175 proteins. The table includes the minimum amino acid length, the maximum amino acid length, the mean, and the standard deviation. All of those two values are included to give you some idea of how conserved that particular protein family is. As with any table in PATRIC, you can click on an individual row, and it will populate this vertical green bar with possible downstream icons. And below it, it will summarize the information for that single row here. So some of the things you can do is you can look at the different identifiers for the proteins in that family. You can see if those proteins are involved in any pathways. It's hypothetical, so they're not going to be. You can download data from that. You could get the FASTA data, either proteins or nucleotides. You can generate a multiple sequence alignment. You can see which genomes have members in that particular family. Note that there were 39 genomes. Here, only 38, so you could look at the members to see who's missing it, and you could create a genome group or a feature group of this. So let's talk a little bit about what we're seeing. You notice that there's 5,097 protein families here. Those are the protein families that everybody shares and all the ones that are unique. This is what's known as the pan-genome, the pan-genome for these 39 genomes. One of the things, if you read comparative genomic papers, often they talk about the core genome. The core genome is those protein families that every genome in your selection has at least one protein annotated in. How do you find that on this table? Well, you notice that there are 3 rows here: present in all families, absent from all families, and either mixed. Either mixed is just saying either presence or absence, we're including it. If I were to click here for present in all families, look what happens to the check boxes. They're all checked all the way down. And suddenly the numbers have changed to 2,286. This is the core genome. So we have the pan genome and the core genome. Another term you'll hear is the accessory genome, which is the difference between the pan and the core genome. Another way you can use this, this table is to find genes or proteins that are unique to individual genomes. So let's click here, absent from all families. And of course, when I click on that, they all go away. But if I were to click an individual genome, and that's saying I want to see all the protein families that this genome has by itself. So if I click on that, you can see the 2 genomes pop up. Can also use it in different ways. I could go in and I can say, I want to see all the protein families that everything that's in these SETI genomes have. Let's see if that works. And this time I'll say they don't, well, let's click on SETI. SETI. See, when you start demanding more of it, it starts getting a little bit tricky. And there's another SETI genome down here. So none of the SETI genomes have everything in common. However, if I wanted to see protein families, so what I was looking for were protein families that all of the SETI genomes had in common. But I could also look at the protein families by clicking on either mixed. I could look at the protein families that all of these genomes, this one F599 is also a SETI genome, that all of them share that the other ones don't. They don't all share them. You notice that some have 2 genomes have it, some have 1. If I wanted to filter on that, you could see the best I can get is this putative lipoprotein that 4 of the genomes have, but the other ones are missing. So you could see that that was part of the problem. But those are ways that you can use this table to see these results. So I'm going to reset it. This will conclude this short video. The next one will talk about using the filters to the filter for keywords and using this functionality, and also generating a multiple sequence alignment from the genome table. Thanks for listening, and join us for the next part of the series of comparative genomic analyses in PATRIC. Bye.